Hi, welcome everyone. Welcome to Next Generation Genomics practical introduction. Um, I'm just going to use this opportunity to introduce myself if you haven't met me already. My name's Nathan Meds. I work at the University of Edinburgh Genomics. I am the training manager at Edinburgh Genomics after doing my PhD at the university in the Institute of Evolutionary Biology. I have been working for Edinburgh Genomics for a couple of years now, and I'm an instructor on this course as well on NGG. Some of you may know me from earlier modules in the course, but if this is the first time, hello, and it'll be nice to meet you guys in the discussion sessions we'll be having soon. So I just wanted to give you a quick introduction to our first practical session, which is online now. We'll endeavour to put these practical sessions up a week before a set of discussion sessions, in which we'll discuss some of the topics covered and answer any questions that you guys might have. Now, apologies this week, we've gone up slightly late, so a day or so less than time than we would like to have given you. That's just due to some technical issues with the server and getting things to work first time around. So I apologize for that. We've had a few teething difficulties, but in future everything will be going up at least a week before, so you have a week to have a go at the practical sessions that I'm about to describe, and then a week later on the Monday we'll have some discussion sessions. Now I'm going to share my my slides with you. So yeah, welcome, as I've already said. And so those discussion sessions will run at 11 and at 4 on those Mondays. The first one being next Monday, the 1st of February. That's week 4 of the course of the semester. And this first session, we're going to be talking about data quality control, data QC. Now, what do we mean by data QC? Well, we're going to go through several steps on this in this task through this practical, and you'll see from the practical guide. The first objective, the first step, is going to be to log yourself into the Bioinformatics 5 server. Now, some of you will already have used this quite a bit already, but if you haven't done so yet, there are instructions to log into the server. You should all have accounts on this server. If you don't, please get back to me or Alex Twyford, and we will sort one out for you as soon as possible. Another way to raise questions is on the discussion forum on Learn as well. So please do get in contact with us there if you have any problems with this. But the first step is going to be to log into that bioinformatics server. We're going to use an RDP protocol to link you in to a graphical desktop. So all the instructions are there, whether you're using Linux, Windows, or Mac. So they should hopefully be relatively straightforward, but if you have any problems, let us know. So the first thing we're going to do is to find where our data is that we're going to be working with. And that data is already stored on the server. To save us all downloading it all separately, we have it stored in a folder called data on the server. And the first thing you need to do is go and make a link to that data. So this will be an introduction for some of you maybe to symlinks, a good way to link to a piece of data without actually having to copy it over into your own personal home folder. We'll then use a tool called FastQC, Fast Quality Control, to assess the quality of these data files. We'll go through each metric that tool uses to assess the quality of our data. And then we'll do some trimming and filtering to improve the quality of our data before we might go on to use that data for further analysis. So this is a very important step. So this evaluation of sequence quality, as I say, is very important because it kind of determines how reliable our results are going to be further down the line. So any filtering and trimming decisions that we make will affect our downstream analysis, whatever that may be. We might be mapping to a reference genome, we might be de novo assembling our reads, we might be doing this would still apply to RNA-seq as well. So we want to make sure that we're making good decisions at this early stage in order to maximize the quality of our data without losing too much of that data. So there's always a balance here between putting it through a very harsh quality control and losing lots of data, or going slightly softer, not losing so much data, but then also not having quite such good quality raw data going into our analysis. Now, this is a balance that it has to be kind of taken on a case-by-case basis, and you just have to use your intuition as scientists to decide what level of quality control is best for your data. And being bioinformatics, we can always go through this step, see what we get out in the end, and then repeat with different parameters and see how that affects our data. So that's we might have to go through this process that I'm about to describe several times in a real experiment to assess what gives us the best results. So before we go any further, I'll talk about the data that we've provided for you to use in this practical. It's a genomic library prepared from the ash dieback pathogen, which is a fungus. Hymen-Scyphus fraxineus, which is it was a data set generated by us here at Edinburgh Genomics and is now publicly available on the ENA, or the European Nucleotide Archive, at the EBI, European Institute for Bioinformatics. If you're interested in this data and you want to find out a little bit more about how it was generated or what the data was used for in the end, you can look at the paper which was published in Nature by Mark Mullen et al. In 2018, and yeah, do feel free to do that. But this is not absolutely necessary. You know, you can take this as a generic data set and just work with it as is. But if you're interested, you can always go there. So what's this FastQC tool all about? Well, FastQC is probably one of the most popular quality control tools out there. There are several others and that can be used, but this, you'll find this to be the most common and one that we'd recommend you use going into the future. So FastQC, the official blurb is that FastQC aims to provide a simple way to do some quality control checks on raw sequence data coming from high throughput sequencing pipelines. It provides a modular set of analyses which you can give, which can be used to give you a quick impression of whether the data has any problems of which you should be aware before doing any further analysis. So repeating myself slightly there, but yeah, you get the picture. This is a tool that allows us to assess quality before going on. Now, one of the key metrics of quality, which you may have covered already, but just to reiterate, because it is quite an important part of quality control of raw sequencing data, are Phred scores. Now, Phred is a quality metric that's very widely used, and it basically gives us a error probability of a base being called wrong. So when we get back our sequence data, if we have our data in the form of a FastQ file, that's a FastA file, but with some quality data, FastQ, that's the Q for quality, we'll have this information encoded within that file. So we don't need to worry too much about how that information is encoded. If you're interested, you can kind of have a closer look at this table, but it's not essential to know exactly how that information is encoded, because we take that encoded information and plug it into a tool like FastQC, which converts that back into a Phred score that we can actually read and analyze. But it's important to know what that Fred's quality score means. So for this table, our central column here, the Fred quality score from 10 through to 50. Now, a Fred quality score of 10 means that we have a probability of an incorrect base being called at that position of 1 in 10, so 90% accuracy. And then we're going up in kind of a log scale, so 1 in 100 is a Fred score of 20, 1 in 1,000 is a Fred score of 30, 1 in 10,000 is a Fred score of 40, and 1 in 100,000 is a Fred score of 50. So the higher the Fred score, the lower the probability of a base being called incorrectly. So a kind of general good standard for Fred score that you'll see used time and time again is a Fred score of 30 as being a cutoff of kind of good quality, a high accuracy of base call. So that's very high, 99.9% accuracy. So armed with this information, we can look at the first metric that FastQC will give us back, and when you run through the practical, you'll find this is the first item in the FastQC report, which is generated as a HTML file that we can look at with our web browser. Now, this plot gives us a Fred score per position within our reads. So we've got lots of reads within our data file, and this is a kind of average quality score at each position within that read. In this particular example, you can see from the x-axis that our read is 40 bases long, so quite a short one. We might find that our reads are a bit longer than this. But this is a good example, or an example of very good data, where all of our positions within all of our reads are within this good quality band. So that's a Fred score above 30. Now, it's unusual when we first come to look at raw data for it to be quite this good. So let's have a look at a bad example, or an example of data that needs something doing to it before we go any further. And in this example, you can see that towards the end of the read, so from kind of base 26, 28 onwards, we really start dipping into this poor quality band. That's Fred scores of below 20. Now, the average here is represented by the red lines within these box and whisker plots. And so the average quality is staying quite high, but you can see the spread of quality across our reads is increasing, and meaning that towards the end of some of our reads, we have very low quality. Now, we can do something about that, and we'll come to how we might address some of these quality issues at the end of this talk. The next metric that we'll get back from FastQC is something called the per sequence quality score. And now this is basically almost that same information telling us how many sequences fall within a particular average or mean quality score. On the left here, we have an example of good data where our mean score for our reads, the vast majority of them, so many thousands in this case, have a mean quality of up in the high 30s, which is great. This would be a perfect scenario. We don't have any groups of reads or subsets of reads that have consistently low quality. On the right here, we have an example of a data set that has a subset of reads that do fall into a low quality category. So despite the majority of the reads being in a high quality, we do also have a group of reads that all have a mean quality that's lower than our ideal, so lower than 20 in this case. So we're aiming for this kind of thing on the left, and we'd be slightly concerned if we saw a whole group of reads that had such low quality. And just to say, if you're interested in looking at some more of these examples of good and poor quality data, you can go to the FastQC website and look at some examples there, along with some more information and more descriptions. The next metric actually looks at something called the per tile sequence quality, and hopefully by this point through Alex's sequencing technologies lecture, you have an idea of how the our flow cells are structured. And actually, when we have a flow cell with covered in lots of clusters, this is broken down functionally into something called tiles, so small areas of that flow cell. And we use this area of the flow cell to assess quality of reads within each section of our flow cell. Now, in an ideal world, we would have a graph that looks like this on the left, which tells us that all of the tiles, all of the areas of our slide gave universally good quality reads across all positions within our reads. Good quality calls within all positions of our reads. But it's likely that we're going to have some lower quality reads present, and it's also possible these might cluster together on a particular position within our flow cell. So if we find that all of our low quality reads were generated in one particular position, within our flow cell, it might indicate that position, that tile, had some problem with it. So we might have a fluidics problem or like an air bubble that was preventing the sequencing machine reading good quality reads, good quality bases at that particular position within the tile. Now, when we go through our kind of quality control step, once we go through our filtering and trimming process of our reads, we may find that these disappear as we trim away bad quality reads. The next metric that we want to have a look at is per base sequence content. Now, this breaks up our sequences into A's, T's, C's, and G's and tells us what percentage of those bases were at any particular position within our reads. And you might have guessed that we'd be looking for a kind of smooth trend over our read, because we've broken our DNA up into random chunks. We would expect it equally likely to be any one of those 4 bases at any particular point, or at least consistently likely across the read. So that's represented here on the left by this good data. So we've got an equal chance of getting a C at any one position, for example. The chance of getting one base over another may differ slightly depending on your organism, but the consistency across our reads is good. On the right here, you can see an example of some bad quality data where we've got a certain positional bias here. So, for example, towards the ends of our reads in this data set, we have a much more likely, it seems much more likely to get a G at any given base position. Now, that could represent some sort of contamination of our data. It might represent some sort of positional bias caused by library preparation, and it's something that we need to address going forward. So the next metric being per base GC content, again, similar lines. We would expect similar concept. We would expect to see a smooth pattern across the length of our reads. So our GC content is likely to be the same at any position within our read. And if we see a peak at any one position, this may represent some contamination with either adapter sequence or potentially some library preparation bias. So the next metric also focuses on GC content, but this is the per sequence GC content. So we're looking at the distribution of GC content across all of our sequences, or across all of our reads in our data, and we would expect this to be normally distributed. The peak of this normal distribution will be dependent on your organism. So as you will be aware, GC content within the genome of any particular organism can change, and you should be aware of this when working with a particular organism, what kind of GC content you would be expecting. And when we come to assess our read quality, we would expect all of our reads to fall within that normal distribution around a peak expected for our organism. What would be slightly worrying in terms of quality control is that if we looked at our GC content within our reads and found that this didn't conform to a normal distribution, we might find that there is a peak at one particular point, or even worse, that there are two peaks. So not represented here, unfortunately, but one potential problem that we could highlight from this graph is if we saw a bimodal pattern. So if we saw two peaks, this might represent the fact that we have contamination with another organism. We might have another organism's sequence within our data. And this is entirely possible. We've seen data like this at Edinburgh Genomics, where contamination with a bacteria has caused a second peak where the two organisms, your focal organism and the contaminant organism, have slightly different GC contents overall, and therefore you see a bimodal pattern. So we want to keep an eye out for that. Another metric we can go by, which is hopefully quite an intuitive one, is the number of ends in our sequence. Now, ends are called when the machine is not sure what base is present, and it's a kind of n, the base hasn't been read. So we want to ideally have a data set where we have as small a number of ends read as possible. And so the good data on the left-hand side shows that there are no ends, or there's no particular spike in ends anywhere within our sequences. Bad data, on the other hand, might show that we have one position within our reads that has a peak of ends within it. So this may be that we've read over the end of a read, or we've got some particular problem reading that has led to a bias in that position being an N. Now we can quite simply trim these off as well. So hopefully we should be able to sort any abundance of N's within our reads out in our filtering and trimming step. The next metric to bear in mind, and this one needs to be taken with a slight pinch of salt, but this is the sequence duplication levels. Now, really good data, like on the one on the left, will show that we have no duplicated sequences. So these are duplicated reads. FastQC kind of uses reads and sequence interchangeably somewhat. So when we say sequence duplication levels, we mean reads that are exactly the same within our data. Now, because these are kind of generated at random and random points within our genome, we'd hope that there were no two reads that were exactly the same. And that's what is represented in this on this left-hand side good data chart, where we have only sequences appearing once. Now, it's possible that we might have some sequence duplication. So a high number of duplicates, so appearing 10+ times, might indicate that we have some contamination. We might have an adapter sequence that continues to be read or continues to make up the entirety of a read. It also might be that we have some sort of enrichment bias. So if we've gone through a PCR step or something within our library preparation, we might have had a tendency to produce more of one particular read. Now, the reason to take this graph or this metric with a slight pinch of salt is that this is taken from quite a small subset of our reads. And also there might be some genuinely good biological reasons why we might have duplications within our data. So it might be that we have gene duplications. We might be looking at an RNA data set where we're looking at the highly expressed genes. So this one is worth taking with a slight pinch of salt, and it's something we can come back and look at once we've done our filtering and trimming step, as with all of these metrics. So overrepresented sequence is another metric, getting towards the end of these. And overrepresented sequences will take any particular sequence that appears multiple times within our data set and display that to us, give us the number of times that sequence is represented, and also some possible sources of that sequence. Now, in good data, we may well have some sequences that are represented many times, but these might well be of biological origin, especially if we're looking at something like an RNA-seq data set where we've got multiple copies of the same gene. But the possible source is not identified, so this leads us to think that these might well be biological in origin. What we're really looking out for is overrepresented sequences that match to a source that FastQC knows about. So built in its memory, FastQC has a list of all of the commonly used adapter sequences that are used to prepare our libraries. Now, there are lots of common ones that are used in general to prepare Illumina libraries, and if we see one of these sequences overrepresented in our reads, then we know that this adapter is being read instead of biological, an interesting sequence. So thankfully, because we know what these sequences are, they're recognized as being a particular adapter sequence, we can take some simple steps to remove these from our data. And the last metric is KMA content. Now, this is a slightly tricky one, but in good data, we're taking, in this case, in FastQC's case, 7 minutes, so see all the possible 7 base sequences and looking for any particular overrepresentation at any one position within our reads. Now, there's good biological reason why we might see some of these short KMAs appearing time and time again in different sequences, but we would expect the chances of finding that particular motif or that particular sequence to be equal across the read. Now, if we find that we have particular KMAs, particular short sequences that occur time and time again at one position within our reads, this might be that we have some sort of contamination. So the KMA content is it's a slightly more tricky one to grasp, and if you want to find out a little bit more about the KMA content metric, then I suggest going to the FastQC website and reading the blurb there. But taken all in all, those are all of our metrics. Now, as we go through these in FastQC, you'll see that FastQC gives us a tick or a cross next to these, whether it thinks that the quality is acceptable or not. Now, we have to kind of use our intuition and look at this whole report in a holistic manner to decide whether our data is of a decent quality or not. We can keep looking at examples of good and bad quality data, but you do have to kind of use a little bit of biological intuition. So have a look at some more examples, and also kind of as you go through the practical, go through these steps multiple times if you need to, and have a look at what effect our next step, our trimming step, has on this data quality. So, as I've mentioned it, the next step, once we've decided, let's say we've decided we have some bad quality data and we need to do something about it, the next step would be to go into trimming and filtering. Now, sequence trimming first off would be to identify any of those adapter sequences, any of those non-biological sequences hanging around in our data, and remove those from our data. We can identify them from a database of known adapter sequences. We can use a search pattern like grep, a search pattern tool like grep, to see if there are any of these sequences, any of these adapter sequences within our data. But we can also plug them into a trimming tool and tell that trimming tool, please remove any of these known adapter sequences. Another trimming step that we can make is to trim any low quality bases off the ends of our reads. So when we look down our read, we can tell a trimming tool we want to take off any bases from the 3' end of our read that fall below a particular quality cutoff. So in this example, 2025, that little red line. This is, although we might decide to use 30, as that is a slightly more common threshold for good quality reads. So we can trim any poor quality base calls off the ends of our reads, meaning that we go from a collection of reads all of the same length to ones that have been trimmed to a variable length because we've taken any low quality bases off the end. Once we've done that, we can then filter our reads. So we can filter based on the mean quality, filter by read length. So if we trimmed low quality bases off the end of our reads and the read has ended up being very short, we could filter that out. This will obviously depend on how long our reads were to start with. And yeah, we can also filter based on the percentage of bases that are above a particular quality threshold within our reads as well. So there are many ways to, many tools available to do this. FastQ MCF, CutAdapt, SeekTK, Trimmomatic. Trimmomatic does what it says on the tin. But for this practical, we're going to use a very widely used and still very popular tool called CutAdapt, which again, clues in the name, is going to cut some of these low quality bases off of our reads. So without further ado, I will let you guys have a crack at the practical session that's online. In that practical document, you'll see that there's bullet points with instructions, black text boxes with command line commands that you can pop into your terminal. We've also highlighted some tips in purple there as well. All this is explained in the document again, along with how to connect to our bioinformatics server. So have a go at those exercises. That I've thrown in a good number of questions there to get you thinking about these quality metrics and thinking about how we might go about quality controlling any particular data in preparation for doing a further downstream analysis. So have a look at those, have a think. Any questions, you can pop them in the discussion forum on Learn, or you can join one of our discussion sessions. So we will be having discussion sessions on Monday at 11 at 4. Please do join one of those discussion sessions and bring any questions along that you may have. But in the meantime, good luck, and I hope you guys have a good explore of this data, and we'll see you on Monday.